Thank you, Jessica, for that kind introduction. Thank you to the organizers for having me here today. As Jessica mentioned, I am very new to UW and to the department, so this is a really great opportunity for me to sort of introduce myself and introduce my work. So I don't have any disclosures. I am a basic scientist, not a clinician. I appreciate that this seminar series has a really wide range of topics, and so I appreciate you guys being here today to really take a deep dive into some animal neurophysiology with me. So my lab and I in general am just very interested in memory and the mechanisms that support memory and how they degrade, as Jessica said. And I think no matter whether you're a clinician or a basic scientist, no matter what part of the body you study, I think we can all appreciate how critical memory function is in our daily lives, to our sense of self, to our ability to live independently and safely. And so it is really imperative that we know how this works and that we can preserve it throughout the lifespan as long as we can. Now, unfortunately, bad news for all of us is that memory does decline with aging, as we all sort of know. This is a good example of that finding. Verbal memory here is the orange line, and indeed it drops throughout the lifespan, and this was really foundational work that came out of the UW aging study here at UW. Now, if this sort of normal cognitive decline with aging is all we face, we're actually the lucky ones, because some of us will fare much worse and will unfortunately face Alzheimer's disease or other dementias, which will cause memory function to decline much more rapidly. Now, unfortunately, Alzheimer's is not at all rare. It affects 6.5 million Americans today, and that number is predicted to grow as baby boomers get older, to the point where now I think the stats are something like 1 in 5 women and 1 in 10 men, or something like that, will eventually develop Alzheimer's disease. As our population ages, as I said, we'll have more and more Alzheimer's cases. And unfortunately, the treatment options that we have for Alzheimer's, as you may know, are really very limited and sort of unsatisfying, despite decades of work trying to figure out some treatment options. I find this plot particularly compelling. Here you see many common causes of death, and over the last 20 years or so, the percentage of deaths from those diseases are all going down, except for Alzheimer's disease, which is like radically going up. So clearly, we are in desperate need of better therapies to face this growing problem. So, one sort of canonical approach in the study of Alzheimer's disease and in general memory function has been to focus on things at a sort of cellular and molecular level, looking at pathology like A-beta plaques and tau tangles, and linking them with behavioral impairments. For instance, an inability to remember the location of a platform in a water maze if you're a rodent, or similar human tasks that show memory impairment. And what I'm really excited about that's sort of started coming in the last few years is more of a focus on the function of networks rather than these more static pictures of pathology. And when I'm talking about network function, I mean the actual measuring the activity of the hippocampus, for instance, and seeing how that evolves as an animal is actually doing something, or as a human is doing something. So the real benefit of this type of approach, in my mind, is that it allows fine timescale linkage of neural processes with their behavioral impacts. We all sort of, I think, can relate to the fact that sometimes you have a quick memory lapse. Usually your memory is fine, but every once in a while something doesn't work and you forget something. And wouldn't it be nice to know what the brain was doing at the time of the lapse to understand what went wrong, what failed to happen in that case? The nice thing about network function also is that it's something you can measure long term and maybe start recording brain activity early before cognitive impairment has really taken over. And you can see what processes start to change early on, what's breaking before the cognitive impairments really become so strong and invasive. I think this can give us a really, let me get rid of this. There we go. It can give us a whole new category of therapeutic targets rather than focusing on, say, small molecules and drugs that are working on a cell and molecular scale. If we can think about new therapies that can focus on rescuing or preserving activity at the network level, this opens up a whole new set of options for us that maybe could be used instead of or in concert with more drug-based therapies. There's also a possibility, and I'll tell you a bit more about this later, that strategies focusing on network function might be more generalizable than drug targets that are maybe targeting one protein or one particular process. If many different disease etiologies end up with the same network level phenotype, then targeting that phenotype might be a really broad and powerful way to help many different many different individuals, regardless of their disease etiology. So, where we will focus on for the purpose of today, and I think it's a fairly obvious choice, is to look at the hippocampus. The hippocampus is shown here in pink in the human brain, as you probably all know. And many lesion studies in both humans and in many different animal models have implicated the hippocampus in being really critical for memory-related function. In addition to that, we see hippocampal pathology fairly early and fairly consistently in many diseases which impair memory, such as Alzheimer's disease. Both of these things suggest that hippocampus is a really good place to focus. So, to dig a little deeper, what are the patterns of activity that happen in the hippocampus that support memory function, and how do they change in disease? So, unfortunately, asking this question in a human is definitely possible, and there's many people doing great work on this front, but it is challenging. We face the problem of non-invasive brain recording methods being often coarse in terms of timescale or resolution of where you're recording signal from. And in my case, if we're really interested in the hippocampus, things like EEG and EEG and fMRI, for instance, EEG, have to parse the signal through all these layers of cortex first. These deep structures are really hard to get good, clear signal from. The alternatives are invasive depth recordings, which go straight to the hippocampus, but most people don't want to undergo that type of surgical procedure unless they really, really have to. So even when you do have an invasive surgical procedure, for instance, in the case of a localization study to find the focus of an epileptic patient, the targeting across patients can be really varied because that implantation is clinically motivated rather than being, research motivated, and generally yields few neurons that we can record from simultaneously. In addition, it's rare to get data from patients who have Alzheimer's disease or who are really old, because often the patients that come in for those types of epilepsy localization studies are younger, and their problem is the epilepsy, not any sort of aging or Alzheimer's issue. Having these types of epilepsy localization studies be your main source of data also limits you in terms of the behavioral tasks that the patients will be doing. They're often bedridden and not feeling great, which means they're not out in the world having experiences the way most of us want to be, and the way we generally rely on our memory to function out in the wild. So for all of these reasons, instead, I've chosen to study memory function in a rat. Some of the studies I'm going to show you are coming from mice, but in general, the rodent brain is conveniently laid out for hippocampal recordings, very nice of them. So what we can do is actually lower probe arrays, whether they're silicon probe or tetrode arrays. We can lower them into the hippocampus, passing through cortex, but it's relatively shallow. We can do this in a chronic prep, which means we can put a hat on the rat, and that prep lasts for many weeks and gives us stable recordings for long periods of time while the animal can be out and about learning tasks and performing memory-based tasks. This approach allows us to record hundreds of neurons in the hippocampus simultaneously. It allows us to look in a range of ages, different ages of rats, as well as disease models. We have transgenic animals that develop symptoms of, similar to Alzheimer's disease. And as I said, we can record during the learning and the performance of a range of memory-dependent tasks, and we can tweak those tasks to test the various memory functions that we're interested in. So, with that background, this technique is what I'm going to use for all of what I'm going to tell you about today. I first want to introduce you to these memory-related patterns of neural activity in the hippocampus that I'm really excited about. These are theta sequences, which relate to memory encoding, and replay as a key neural mechanism of memory consolidation. I'll tell you a little bit about how these patterns of activity are affected in models of Alzheimer's disease and in aging. And I'll introduce to you a novel strategy that I've developed during my postdoc, which allows us to promote or manipulate replay. And we'll go into some future directions. So the first thing you need to know really about hippocampal activity in behaving rodents is that we find place cells in the hippocampus, and these are hippocampal neurons which are tuned to spatial location. This means that as an animal wanders around a box, a cell will fire reliably in a certain location. And if you look at the firing patterns indicated by the heat map here of different cells will fire in different locations of space, such that between all these cells, you're sort of tiling the environment with individual neurons in a unique way. And this discovery by O'Keefe and his colleagues in 1971 was so impactful for the field that it garnered the Nobel Prize in 2014. And what we now know is that about a quarter of CA1 principal cells act as place cells in any given environment. If you choose a different environment, a different 25% or so will serve as place cells there. And the spiking of these neurons, the key characteristic that makes them place cells, is that they are spatially restricted, so they have a single or a few restricted place fields. It is environment specific, such that they would fire differently or not at all in a different environment. And it is reliable, such that if you put a rat back in the same environment, the same place cell will fire in the same location fairly reliably. And in general, we study these place cell activities while the animal is moving. We'll get to what happens when they are still in a little bit. So all of this put together means that when we're recording from ensembles of neurons in the hippocampus, we can plot, 13 individual neurons here, ordered by where their place field is as the rat runs along this trajectory, say on a linear track. And this ensemble of cells will fire sequentially according to where their place field is on the track. Collectively then, this whole group of cells, this ensemble, represents the experience that the animal is having as he's running along the track. And the fact that we can rely on these hippocampal neurons to represent space robustly is a huge point of leverage for us to understand in general how the hippocampus maps experience and how that experience gets stored as memory. Because now we have this reliable mapping between the spiking activity that we observe and this very accessible behavioral variable of position. So one of the things I want to propose in general and leave you with is this idea that we can leverage all that we know about how normal learning and memory works and see how this is going awry in the case of aging and disease. So one thing that is fascinating, I think, is that if you zoom in on what's happening as the animal is running around in its environment, you actually see that there's even more organization and structure to this spatial firing than you see if I just showed you the zoomed out view. So if we zoom in, stretch the time axis here, you'll notice that as the animal is running through space, first of all, there's this very noticeable large amplitude fluctuation in the local field potential that is called the theta rhythm. Now, if we break up the spiking by individual cycles of the theta rhythm, for instance, let's focus on this one, you'll see that the cells that fire during this theta cycle also fire in the order that the animal, in the order that they sort of are laid out on that track. So that even within this 125 millisecond, let's say, theta cycle, the animal gets a whole sweep of activity basically representing the whole track, or at least a region right behind where the animal is, going through its actual position to going right in front of the animal. So this is a little time-compressed little slice of experience that goes from just a second ago to where the animal's going. And these sweeps of activity are called theta sequences. And they're thought to be really useful because they bind, you can think of them as information packets that bind this little chunk of experience together by having these cells all fire in quick succession. So this is binding spatial information, probably other sorts of non-spatial information about the experience, all together in a little packet. So that's what's happening as the animal is moving around, and that's how we can think about encoding an original experience that the animal is having. What about when the animal is still, and how does that experience get saved? So the phenomenon that I want to introduce you to here is called hippocampal replay. And this is the phenomenon that we notice when an animal is still. Every once in a while, again, the time axis here is really zoomed in, you see the same group of cells, that ensemble that corresponded to the original experience, will reactivate in a really time-compressed manner, even though the animal is sitting somewhere totally different, doing something totally different, maybe asleep. It'll have this reactivation event of the experience that it formerly had. And this is what we call a replay. These replays happen to coincide with a very characteristic oscillatory burst in the local field potential, and we call that characteristic activity a sharp wave ripple. And indeed, you'll hear a lot more about those for the rest of the talk. But we consider this the LFP signature of replay. And as I said, the replay is what we're talking about in this sequential reactivation of a group of cells that recapitulates an experience. And these things happen when the animal is still, and that can be still when it's sleeping or still when it's just pausing and taking a break from its active behavior. That's when these things pop up. Now, within the hippocampus, sharp wave ripples are initiated in the CA3 subregions here at the ends, looking bilaterally on this coronal section. But they engage hippocampal ensembles that span both hemispheres. So there's a massive amount of coordination taking place during these events to organize this sequential spiking across hemispheres and between CA1 and CA3, as well as coordinating with the rest of the brain too. Now, this reactivation event, the replay, is thought to be really important for facilitating the consolidation and integration of this experience throughout distributed brain-wide circuitry. And we know that it's important because several manipulation studies, as well as correlational studies, have shown that if you disrupt ripples or replay, for instance, you see impacts on learning and memory performance. So we know these things are really critical. Just to bring it back to humans a little bit, sharp wave ripples are actually quite well conserved. They can be detected in human medial temporal lobe recordings. Again, these are often from those epilepsy localization studies. Here's a good example. You can see the ripple itself in that LFP trace. And this study showed that spiking single neurons in the medial temporal lobe are also modulated by the ripple event. Studies like this, however, can't really get to the content of replay or figure out what experience is being represented because we can't really make that decoding model. I'll come back to that. So we do know that human sharp wave ripples do appear to be modulated by memory-dependent, memory-engaging tasks, suggesting that they are playing a similar role in humans as they are in rodents. And we have some evidence that they do coincide with replay. We can get a little bit to, is there content? What is the experience being replayed? Using some MEG techniques. So moving into the disease case, back to rodents again. This is going back to my PhD work, actually, when I was studying ApoE4, which is the major genetic risk factor for Alzheimer's disease. And I was working with ApoE4 and ApoE3 knock-in mice. Now, the ApoE4 is the risk factor allele. And when you replace mouse E with ApoE4, these mice show striking differences in their sharp wave ripple compared to E3 knock-in mice. E3 is the neutral allele. These changes include, first of all, that there's fewer sharp wave ripples overall, and the remaining sharp wave ripples that the animals have are abnormal in various ways. For instance, their duration, their frequency of oscillation, and the oscillatory components that occur alongside the ripple. And you can just see the most simple, the most basic finding from our original study is just that the number of sharp-wave ripples in these E4 knock-in animals is just much less, much reduced compared to the E3 animals. A follow-up study that we did also showed that these abnormalities in the sharp-wave ripples in early, sort of middle ages was predictive of the severity of behavioral impairments that these animals would go on to develop. And so that was really exciting to us because here we have an early predictor of later decline. Maybe this could be a really powerful biomarker for us. Now, excitingly, around the same time that we were working on this in an ApoE4 model, other labs were looking at other types of Alzheimer's disease rodent models, for instance, A-beta overexpressors or tauopathy models. And what was very cool, actually, was that we all sort of arrived at the same conclusion and all found that these models had a reduction in their rate of sharp-wave ripples. So this was a very interesting suggestion that there's maybe a common failure mode of the hippocampal network in this sharp-wave ripple sense, and maybe that means that the sharp-wave ripples could serve as a compelling therapeutic target that would be relevant for a wide range of disease etiologies, kind of as I foreshadowed a few slides ago. Okay, so what about in normal aging? Because of course all of these Alzheimer's models, or normal Alzheimer's, takes place on a background of aging. We also have some evidence that sharp-wave ripple abnormalities would occur in the case of normal aging, although there's been less done on this front. And we do have some evidence that place cell features are altered with age, such as reduced reliability of your place cells, that kind of thing. So some really big questions remain, and these are really the gaps that I would love to fill with my own lab here. First of all, how do Alzheimer's and aging impact replay, particularly replay content? A lot of the studies that have come before have focused on sharp-wave ripples, but I really want to dive deeper and get lots of single neurons so we can see what that ensemble is doing. How is the content of replay affected here? And also try and pick apart whether the Alzheimer's and aging are having similar phenotypes that are just exacerbated in the case of Alzheimer's, or if they're really distinct ways that the hippocampal network is changing. So it'll be nice to have both questions going in the lab with the same behavioral tasks, for instance, the same methods. We can really compare these things head to head. So to do this, as I sort of alluded, we're going to use aged and Alzheimer's transgenic rats. We are going to use complex memory tasks, which I will tell you about in a minute. And we're going to use some computational approaches which allow us to sort of analyze all that single neuron data and tell us more about the content, what is being represented in these animals. So first, to introduce you to the task. This is a task that I developed during my postdoc, and it extends on what I saw as a limitation in the field, which was that many behavioral tasks were quite repetitive and simple. And I wanted something which was complicated enough that I could really disentangle the link between replay content and what the animal was doing. And that's how I came to this funky looking maze. This is a large environment. It's about 1.5 meters by 1.5 meters. It has walls. The animals run around it. I'll show you a video in a minute. But these rectangles here are reward ports where the animal can poke its nose in, trigger a beam break, and receive a drop of milk if it's doing things in the right order. The goal of this task, or the structure of the task, is such that each trial in the task is initiated by the animal poking at home. It then has to choose whichever of the center ports lights up, poke its nose in there, and then it gets presented with a choice. All of the 8 arms light up, and the animal has to choose one of the 8 arms to visit with no cues. And at any given time, only one of those 8 arms is going to be the goal location. So over many trials, the animal has to try one of the arms, figure out which arm is actually rewarded, remember that location, and then continue going there. Every once in a while, the goal location changes though, so the animal will go to where it thinks the reward is, it will be disappointed, and then it will go back to searching among all of the arms to find the new goal location. So here's a top-down video showing what that behavior actually looks like. To orient you, this is the rat. It has an LED, a red and green LED mounted on its head. This is just the reflection on the wall. Don't get confused by that. And you'll see the various ports light up as the animal pokes there. This video is 4x or 5x sped up, so they don't actually move this fast, but just for the sake of time. So you'll see on this trial, the animal is searching for the goal location. He doesn't know where it is. He just tried arm 4, did not get reward. Now he's going to try arm 6, still no reward there. Now he's going to happen to choose arm 2, and to his surprise, he gets reward. So he'll spend an extra second consuming that reward. And then you'll see on this next trial, he remembers exactly where he needs to go, and he heads back to arm 2 because he remembers that that is the goal arm. So that's how this task works. It's turned out to be really, really useful, and it's what I plan to use in the lab, at least to start out with. So I do want to study aging, however, and it was important that any behavioral task we want to use shows an impairment in aging so that we can then sort of drill down on what the neural activity is during this task that's causing the impairment. So in a pilot study that I did a while ago, I put 2 young and 2 aged rats through training in this task. And what I found, although of course there's a lot of variability, is that in general, the aged animals in gray here had a lower fraction of goal arm visits than the young ones, suggesting that they are less good at remembering where that goal location is and getting themselves there. There's lots more characterization we'll do on the behavioral front, but that at least showed me that there's something to go and dig into using this behavior in the old animals. Okay, so the other thing I wanted to introduce you to was the computational methods that we're going to use to analyze these neural recordings. What we really want to do is decode replay content, so understand how that spiking relates to the experience that the animal has had. And the way we do that is to build an encoding model based on the spiking that we record as the animal is moving around. So if we collect data as the animal is running around, we can make a mapping of basically this cell in red tends to fire at this location. This cell in blue tends to fire at this other location, and basically make a big model that maps spikes to location. We can then use that model to decode spikes that we collect when the animal is still, because this is when we know there might be replay events where the neural representation is representing something other than the animal's current position. So we can take the spikes that we observe during immobility, and especially during sharp wave ripples, and estimate the neural position that we think is being represented based on the model that we built during movement. So a lot of this I've done in collaboration with a former colleague, Eric Denovelis. I won't get into the details of the algorithm, but I'm happy to talk about it later if you want to find me and ask. But I do want to say that we're doing clusterless decoding, which means we don't have to explicitly group cells by their neuronal identity. We can just take features of the spike waveform and relate that to position instead of cell identity. This saves a lot of time, and also saves a lot of arbitrary decisions on our part. We also layer on top a state space movement model, which allows our prediction of position to incorporate estimates about where the prediction was in the previous time bin. But it's also a fairly flexible model that doesn't hold the position to be continuous in time. Again, we won't worry about the details too much. All of that to say is that these approaches allow us to produce videos like this. So what I'm going to show you here is the decode corresponding to the time during a ripple event. The animal's actual position is shown here in magenta. So you'll see that it's sitting very still at one of those center ports. But the neural representation of position is shown in green, and you'll see that that does not stay quite so still. So as I play the video, the animal's not moving at all. This is about 200 milliseconds of data. And you'll see that the neural representation of position takes a beautiful run down arm 5, even though the animal is actually staying completely still. And this is a classic example of a replay event, a replay of a run down arm 5. So this is a little bit of new data to show you. I unfortunately don't have any quantifications, but this is what I'm working on, and I'm excited about it. We now can do decoding from one of our old rats that we've recorded. So this is a 32-month-old animal. This is very old, 70 or 80 in human years. And I'm just going to show you the decode of a time stretch when the animal is just running or walking down. Oh, and I should actually show you the animal's position, just like the previous plot, is shown in red here, and then the decode, the neural estimate of position, is shown in green. So if I play the movie, what I want you to appreciate here is that in general, the animal is walking down the arm, and the neural representation of position fairly tightly maps onto the animal's actual position. And the reason I was excited to see this is because there was a possibility that in the old animals, their place cells could be so unreliable and messy that we wouldn't be able to decode position as the animal was running along in the first place. But that's clearly not the case. We still have enough spatially modulated firing that is reliable enough for us to decode the animal's position. So spatial location is still represented in the age graph. This is good. Lots of things to do next. For instance, quantifying the precision and the reliability of the spatial representation as the animal is moving around. Investigate theta sequence organization, like what I introduced to you before, seeing if we have these nice sequences packed into each cycle of theta. And understand how the neural estimate of position might vary with the animal's behavior. For instance, when the animal makes a mistake, does it think that it's running down the right arm, but it chooses a different one? Or does it just completely lose a representation of where it thinks it is at all? So of course, we also want to look at replay in these old animals. This is the same old animal, and this is now a replay event. So this is much slowed down time scale. Here the animal again is sitting still at one of those center wells. Give it a sec, and you'll see two nice replay examples here. So a quick run down arm 4, and then back. I promise there's another one coming. Here we go. This time he's going to replay a run down arm 7. So what this tells me is that, okay, we still can find examples of good replay in aged animals, but of course there's a lot more to quantify. For instance, the precision and the content of replay, which arms are being represented when. Not all replay events are interpretable. The ones I showed you are nice, like clearly it's a run down arm 7, right? But the example I'm going to show you here is a replay that is much, much messier. And we know this should be a replay because we see the LFP signature, that sharp wave ripple, but you'll see here that the neural representation is just sort of flickering everywhere. Now, I've seen events like this plenty of times in young animals too. I'm not saying this only happens in the old animals, but one thing I'm really looking forward to digging into and quantifying is how often you see things like this in the aged animals or in the diseased animals compared to in the young animals, and when that tends to happen relative to the behavior that the animals are doing. Okay, so returning to the outline I presented to you at first, I've introduced you to these memory-related patterns of activity, the theta sequences during movement, and the replay as a key neural mechanism for consolidation that we see during immobility and sleep. And I've mentioned how these might be affected in the case of Alzheimer's disease, and in aging, at least in the rodents. So now I want to introduce you to this main project that I completed as a postdoc, which was developing a strategy to actually promote replay. So to sort of recap, why would we even want to do this? To really spell it out here, if aging or Alzheimer's disease, for instance, reduces or impairs replay, and if those changes are contributing to a failure of memory consolidation, then it follows that if we could increase replay or rescue those abnormalities, we might have a chance of boosting consolidation, and maybe that could lead to improved memory performance. Those are the hypotheses to test here. So what would an ideal manipulation actually look like? So one, we would want to achieve significant and specific modulation of sharp wave ripples. We don't want to just broadly turn up excitation in the hippocampus, for instance. We want to make sure that any manipulation we do preserves task-relevant replay. I don't want to just get sharp wave ripple events that have nonsense content, right? I want the animal to really be replaying real experiences that are relevant to the behavior that the animal is doing. I also want to not disrupt behavior. Of course, ideally, we would improve it, but we'll get there. And we want to use a technique that has the potential to be clinically translatable. So others have, of course, been working on these types of things. And one way this has been done in the rodents, at least, is to use optogenetics. So to infect cells with a construct that makes them optically responsive, and say, turn up the excitation during a ripple to extend it and make that ripple event longer. And this type of manipulation has been seen to be useful. But because it depends on this exogenous construct, it's hard to think about translating that into humans. I wanted to see if we could take an approach that would be a little bit more amenable down the road to translation. So the idea that I had, and that I pitched to Lauren as this crazy thing we could try, was the idea of using neurofeedback to try and manipulate sharp-wave ripple events. The way this would look in our hands is that we would be detecting sharp-wave ripples in real time as the animal is going about its behavioral task, or at certain times when we're asking the animal to produce ripples. When we detect one, we would lock that to, or rapidly respond with a sound cue and the delivery of a food reward, which is unambiguously positive for the rat. Basically saying, good job, yes, you did a ripple, okay. And the question is, what can this do? We implemented this neurofeedback paradigm in the task that I just introduced to you, which is a task where we understood how the behavior worked and we understand what replay content generally did. That's an important sort of comparator for us. And here's how that actually looks. So those two central wells that in the initial introduction had no difference between them now end up having different rules. So at the left-hand well, these are now going to be neurofeedback trials where the animal has to go to that port, hold its nose in the port and just wait until a ripple is detected, at which point I'll give the sound cue and deliver reward. This looks something like this, monitoring the activity, the LFP, and then when we detect a ripple, the animal gets its drop of milk. On the other randomly chosen 50% of trials, these are going to be delay trials, where the animal has to go to the other port, hold its nose in the port, and perform roughly sort of externally the same behavior, just wait there and be still. But here, the trigger is not, the reward is not locked to the detection of a ripple, it just happens after a certain delay. So even if the animal has ripples during that delay period, it doesn't matter. He still has to wait for the time to be up, and then he gets his sound cue and his drop of milk. Now, what was key here is that early in training, we make the task really easy, such that the animal just has to have a tiny ripple. These happen pretty frequently during immobility in order to get the milk and continue with the task. But over 2 weeks of training, we raise the threshold for ripple detection such that the animal has to produce a larger and larger ripple event in order to proceed. So now we're asking for something that occurs quite infrequently. We're making the task harder and harder and forcing the animal to really learn to do something here. Okay, so here's what they learn to do. This is looking at the replay rate, just to introduce this plot to you. We've got replay rate on the y-axis and time locked to when the animals get that sound cue and reward delivered. And this is first plotting a control cohort with no neurofeedback training. They just have to wait a certain amount of time, then they get a reward at the center wells. They can consume their reward for a few seconds, and then they go on and make their choice. And you'll see that this animal, representative of all the control cohort, has very few ripples during the time when it's waiting. And then after it gets its reward and it's consuming its reward, it has a whole bunch of ripples. So then we see the ripple rate go up. In contrast, the manipulation cohort animals, after their several weeks of training, show really striking changes in when they're having ripples during this period. So especially on neurofeedback trials, plotted in red, the animals have a very high rate of ripples even during the time when they're just waiting before the reward is delivered, and then a slightly lower rate afterwards. And this is even affected on the delay trials. So the animals have clearly learned overall that now they have ripples more often during this delay period than the control cohort. Yes, so it's a lot to look at, but basically we shift the sharp wave ripples from mostly being after a reward to mostly being before reward. And I can show you this quantified over our cohort of 4 animals in the neurofeedback cohort and 4 animals in the control cohort. And you'll see that the sharp wave ripple rate during this pre-reward period is highest for the neurofeedback trials, sort of in the middle for the delay trials, and then very, very low for the control animals. So we've really shifted when they're having ripples here. So what about replay content? I mentioned that any manipulation that I would be excited about also has to preserve that nice replay content. And indeed, we saw that it does. We can take advantage of the fact that these trials, that this task has the animal visiting many different arms across different trials. And so we can unambiguously say, how is replay modulated by the behavioral context of the arm? For instance, are the arms that the animal has just visited more or less likely to be replayed, for instance? And this is going to be a big plot, so let me introduce you piece by piece. What we're showing here on the y-axis is how the chance of replay is modulated by the arm that is replayed. So for instance, the immediate past arm, what we found in this task, is that the immediate past arm is less likely to be replayed, which is why these values tend to be below 1 here. And what you'll see through all of these categories is that the control animals and the neurofeedback animals tend to be really similar. So we haven't changed what gets replayed. We found that the future arm has a slightly increased rate of replay. So you see most of these values are just above 1, and that's true for both cohorts. And then what was a striking result for us from a previous study is that in this task, the animal is most likely to replay the previous goal location, not where it's about to go. And we found that this bias was preserved in the neurofeedback animals as well. So essentially, this plot just shows us that the content of replay shows the same biases based in behavior that we would expect in the control animals, or that we saw in the control animals. So even though we're having more replay overall, how it gets parsed out between the behavioral allocation is unchanged. So to sort of summarize this section, what I've shown you here is that this neurofeedback approach can train subjects to achieve a more ripple-conducive state, and that during this state we have replay content that matches the types of content we would expect. And it doesn't, I didn't show you this, but it does not disrupt the animal's behavior. So this neurofeedback paradigm didn't prevent them from learning or performing the task. And this tells us that the subjects can actually learn to modulate this memory-related pattern of neural activity, which is very cool to see. But you're probably wondering what happened to the behavior. I told you it didn't mess it up, but we also didn't see that the behavior was enhanced in these animals. And I think there's a few reasons why that could be. First of all, we were testing this paradigm on young subjects. So they're already performing the task quite well. They have plenty of ripples and replay. Having extra probably didn't actually make a difference for them. In addition, we made the choice of having trials, neurofeedback trials and delay trials interleaved. And in the few years since we designed this study, I think we've come to an understanding that the consolidation processes that replay are really helping with are happening over much longer timescales. So because we interleaved the trials, we might actually have sort of diluted the overall effect on behavior that we could have had, making it hard for us to see a difference. What I want to do going forward is move to a block structure, where say you have a whole block with a single goal location that the animals have to learn while they're having more ripples. And then a whole block that follows, counterbalanced of course, where they would have fewer ripples and learn a new goal location and kind of compare over longer timescales what effects on behavior we might see in those cases. We also were working with a task which doesn't force the subject to remember a goal location for long periods of time, for instance, from one day to the next. And I think extending the period, the memory burden that we are forcing on these animals would actually allow us to pull out more nuanced effects of the ripple manipulation as well. So clearly we have a lot to do, and I'm excited to get into it as I sort of build my lab here. As I had mentioned, there's lots of ways we can improve the task to more sensitively understand how replay might be contributing to behavior and how a manipulation could affect that. I also, of course, want to take this neurofeedback paradigm and move into, say, the aged subjects or a disease model when the replay is, or the sharp wave ripples are likely to be impaired. Because if they are missing replay, if they're having not enough replay and we can boost that back up to normal levels, maybe that's where we're going to stand a much better chance of seeing a behavioral impact. As I mentioned more towards the beginning, we also have a lot to do in terms of deeply characterizing the nuanced changes, or maybe the very obvious changes, that we would see in terms of replay and theta sequences throughout aging, middle age, as well as these very old animals, and through the course of disease progression using a transgenic rat model. In the very long-term sort of like ideal case, I would also love to collaborate or explore the idea of using a neurofeedback intervention like this in clinical populations. This doesn't require any sort of exogenous construct like optogenetics. If you have access to hippocampal recordings, you could easily build an app, for instance, gamify it that the human gets a gold star or something when they have a ripple, or when ripple rate is modulated positively. So anyway, I think there's lots of interesting ways we could think about trying to test this type of approach in humans to see whether human patients could actually learn to modulate sharp wave ripple rate, access a state that was more sharp wave ripple conducive, and see whether that could actually have cognitive benefit for them. So with that, I will wrap up. I have lots of acknowledgements at this point because I've shared with you a slew of all things all across my training. But I will say I'm really grateful to be here at UW. I have one lab member, Ian. Thank you for coming. We are recruiting, so if this was interesting to you or if you know folks that might be interested, please check out our website. I'm also really grateful for biostructure Department of Biological Structure and DLNP for really helping us get set up. My postdoc training, my mentor Lauren Frank, and the lab there at UCSF, especially my collaborator Eric and my technician Danny, helped with a lot of the data that you saw today. Various open source tools that have come out of the Frank Lab are going to be really instrumental for me going forward. And then also have to acknowledge my PhD mentor, Yudong, for all that ApoE4 work that I shared with you today, and his lab also at UCSF, and our funding sources. So I thank you for your attention. I guess this is when I put up this slide. Please fill out your forms or whatever. And with that, I'm very happy to take questions. Yes, Jeff, go ahead.
Thanks, Anna. That was great.
Yeah.
So, you're the only person in the department who records live from rats, I'll have to say. But we do a lot of complementary things in both pathology and biomedicine. I was wondering just some thoughts on potential ways that we could contribute. One question was, when you put recording electrodes in and you record from, say, the place cells, after you take them out, can you go in and figure out which one was which and to do histology to biochemically or immunochemically characterize that cell? And then the second question is
Can I answer your first one first? Because I'll forget otherwise. Yeah. Okay, so right. In terms of thinking about areas of overlap between all the human pathology stuff that you guys have and what I could do, could I take a slice of tissue, for instance, after doing these recordings and figure out which neurons I was recording from and characterize them? With these in vivo recordings, it's really hard to think about how we might do that at a single cell level. One thing I have in the works right now is a collaboration with folks at UCSF, Salvieda's group, which specializes in RNA-seq. And so what we're doing is we're taking samples of the whole CA1 after doing these recordings and sequencing that tissue from aged animals, trying to see broadly how are those cells changed in terms of expression profiles, and see if we can link that to the neural activity phenotypes and to the behavioral phenotypes. I think if you wanted to do something more like what you were saying, an electrophysiological recording is harder, but an optical recording setup might be easier, because then you can at least label subtypes of neurons and see what their characteristics are later. We could totally do that. It sort of depends what neural patterns of activity you want to see, because with optical techniques, it can be very hard to do things like replay analysis because you don't have the temporal resolution of single spikes. So I think there's definitely potential for overlap, but we just have to figure out what level of specificity we have that at. Okay, second question.
The second one, on the lab medicine side, we are pretty good at getting increasingly better at measuring the concentrations of biomarkers in both tissue and the cerebrospinal fluid, the extracellular fluid. In the work that you cited, or maybe your own work in models of Alzheimer's that say have tau or A beta changes, are there dose response curves? So the more tangles or the more plaques, the worse the tracings look, such that quantitative analyses of biomarkers might actually be informative and complementary to what you do?
Yeah, that is such a good question, and I know that is not known. I think we're at the... Oh, yes, thank you. The question is, my interpretation of the question is, can we actually use blood biomarkers fluid biomarkers to predict what might be happening in the neural activity level? And I think if we do really want to push on the idea of these neural patterns of activity like sharp wave ripples being a good biomarker for disease progression, it would be fantastic to be able to validate them with fluid biomarkers. And then the fluid biomarkers, if they are related, are way more accessible than anything you would get from neural recordings. So if those did relate, it would be very cool. I don't know of any evidence that suggests that they would. I'm also not sure that they would, given the fact that there's a wide range of, say, amyloid burden and a wide range of cognitive impairment, right? Like there's a disconnect there. And I hope that the neural activity patterns are more correlated to the behavioral impact, which might mean that they're less correlated with the amyloid burden. But I don't think we know, and I think there's a lot to tease apart there. Yeah.
Thank you. That was a really great talk. One of the main differences when I think of rodents and humans is the rate of adult neurogenesis. So how does that play into this phenomenon that you're studying? And then how do you speculate that that's contributing to the mechanisms of how you want to look at this in neurodegeneration in humans?
Oh, yeah, that's a great question, and it's something that I don't think about very often and that you and I should talk about for sure. The question is, how does adult neurogenesis and differences between rats and humans in terms of their adult neurogenesis play into the types of neural activity phenotypes that you would see? What I do know, for instance, in the APOE4 model, there are changes in neurogenesis in that model. But again, if that sort of doesn't translate to what's happening in humans, it kind of doesn't matter. I don't think a lot of this stuff has been looked at in terms of linking, say adult-born neurons and their contributions to sharp wave ripples, for instance. That level of interaction is really not known. The answer is I don't know, but we should talk. Yeah. Yes.
Some questions online.
Great.
Do theta sequences during sleep look the same as in awake rats? And is there a temporal association with REM sleep or other forms of sleep and theta ripples, or reinforcement of theta wave patterns during sleep?
Okay, so the question is, what do theta sequences look like during sleep? So theta sequences we generally think of as an awake phenomenon only. We do see theta power during REM sleep, and then we see sequential activities that is more similar to what you would see during movement, during awake movement. how that is affected in disease and aging, I don't think we really know. Did I answer the question there?
I hope so.
Okay.
That's not my question.
That's, yeah.
And then one other question I'd like to. Rachel Sheridan asks, she says, fascinating presentation. Has there been any investigation of the association between spatial replay and auditory memory?
Ooh.
The example is, could the replay be the reason that I associate certain songs played in the car with locations that I hear them?
Interesting. So I think the question is, what do we know about sort of auditory replay, and do we think that it might work the same way as what we see with spatial replay? And we tend to use space and spatial decoding and replay because it's a very convenient behavioral variable to track, the physical location of the animal. But I think a lot of what we learn about how the hippocampus, compresses experience in terms of replay, all of what we see in the spatial modality, I think, is likely to generalize to other modalities of experience. So it's not just that you're performing a spatial trajectory. You also have the sound cues of walking around, the smells of the room as you move. And I think all of those different modalities of experience are probably wrapped into the replay the same way that the spatial information is wrapped in there. It's just that we don't usually build our decoding models with the information about other sensory modalities to be able to decode what's happening during replay. So I think there is likely auditory information in those replays, but it's hard to prove that. We know that there are auditory fields or cells that respond to specific sounds, for instance, in the hippocampus. That's been shown. But not to the level of having a replay of auditory sequences, but I expect it is there.
This question is for me.
Okay.
So it seems like with your SEM, trying to stimulate the rats to do the SEM waves.
Sharp wave ripples?
Yes, the sharp.
Okay, yes.
Are you really just asking the rats prompting them to remember where they've been walking along and replay in their minds where they've been and how they've been moving along those paths?
So one of the things that I really was actually worried about as I implemented this paradigm was I didn't want the animals to be able to solve the task by just replaying a certain thing, right? If they always learned let me spout off a replay of breakfast and somehow that just triggered off the same cells every time, that wouldn't be useful to me, I think. That's the animals learning to do something to cheat the task, basically. I wanted to make sure that the replay that we saw spanned the gamut of all the different experiences that they're having. So it was comforting to me that we didn't see replay of, say, one particular experience overloading the system. I do think we are getting them to learn to be in a state that is conducive to ripples and replay. Not any particular replay, but just like a calm, replay-friendly state. I think because ripples are quite short, I'm not sure if I actually showed this, but they tend to be on the order of about 100 milliseconds, and that's pretty much below what we would think of as something that was consciously detectable. So I don't think the animal is consciously aware, and I don't think a human would be consciously aware of, say, having a replay of this particular experience. I think it's all sort of subconscious. Of course, that's hard to prove in a rat. So, yeah. Okay. There's one more? Okay.
If you have time.
Sure.
So are the anatomic location of the space neurons within the hippocampus same for different animals? Connection between neuroanatomy and environmental space?
Yeah, so place cells are actually not topographically organized. So there's cells that are tuned to different spatial locations, scattered all throughout the cell layer in no systematic way. So you wouldn't, for instance, be able to predict the spatial firing of one neuron based on recordings from another animal. There's not a predictable structure there that we know of. Okay. Thank you all very much for your questions and your attention.